Okay, so what we're going to do now for the next 2 hours is we're going to have members of the fire team come up and talk to you about the work that they're doing under the fire initiative. And I believe you're going to give a little overview of the fire initiative.
No.
No? Okay, so let me do this instead then. We decided in talking with Ed and some of the other scientists and thinking about some of the programs at the NIH that are very successful. One of the programs at the NIH that I really like is called a program project. And what that is they ask 4 or 5 researchers to come together and put together one single grant that says, the 4 of us are going to work together. We're going to work together on this one single topic. We're going to each bring our expertise to it. And part of that program project is you have to say that you're going to meet regularly and you're going to talk to each other. And we thought, wouldn't this be awesome to do in Angelman Syndrome? And Ed was willing to spearhead this for us, and we have an amazing team, and this year we got to add to it, so you're going to hear our new fire investigator. But the point of this is for them to be able to do the basic research that they can do to help us get to the point the pharmaceutical companies can then take these things and run with them. And you have already been hearing that Ed does a lot of preclinical work for some of these groups as well. But we didn't want them to miss the opportunity to hear something that happened, let's say, in Ed's lab on Wednesday that could really affect what David Siegel decides he's going to do on Monday. That kind of close ties, close conversation, that's what makes science go forward very, very rapidly, and that's what we wanted. So now you're going to get to hear each of our fire team members tell you a little bit about what they've been doing. And I, again, feel like we don't really need to introduce Dr. Weeber. So I'm just going to say that he is, of course, a superstar in our community. He's also the head of our scientific advisory board, and he helped put together the fire initiative and has run it. And everyone can clap because the Australian police found all of his stuff, and he's able to come here and give you a talk.
What does FIRE stand for again? Could you just say it again?
Fast Integrated Research Environment. Thanks.
So back in the old day, and I think that there's probably some people in here that can appreciate it, whenever we give a presentation, we used to do this on carousels with slides. And so you'd have to have your talk done, weeks in advance so that you could get your talk together. All the slides would be in the right orientation, and you would carry these big carousels around. Now with the advent of PowerPoint, I can see all the fire team people working on their talks really early in the morning and on the plane ride down here. So especially for me, yes, I'm sure some of you heard about my adventures in Australia. I had a great time in Australia, and I know there was somebody here that just said they were from Australia. Yeah, it was an unfortunate incident, but I recovered everything. The police there were fantastic. They went after the people that had stolen it out of my the room that day in Cairns, and so I'm here with you today, and I can show you some of the stuff that we're working on currently. Yeah, I tell my students this all the time. It's harder to give like a 10 or 15 minute talk than it is an hour-long talk, because you just don't have time to get into everything. So I'm just going to kind of hit an overview of some of the things that we're doing in the laboratory, some of the things that we're working on, and most of this are some of the subjects that we're working on that are close to publication or we've already submitted for publication. So you can see we're moving quite along. So. I have this slide up because just to give everybody somewhat of an overview, and I actually wrote a review article with my graduate student, Stephanie Charlone, that this figure is going to come out with, but it kind of gives an overview of this idea of where targeted therapeutics can go, and we've talked about this upstream or downstream. And this really kind of shows you that you can target a lot of different areas in the synapse. If we're looking at this at a molecular level, this is a picture of a synaptic connection. We've got, you have the nucleus up here where the gene, the UBE3A gene is. These are synaptic connections. These are inhibitory connections. So when we start talking about upstream or downstream targeting, we're really talking about either targeting the gene where the disruption is, and we know where the gene is, we know what the gene is, or we're talking about trying to overcome the absence of the gene, either through what's being talked about earlier today with Ovid and Agilis and disruptive nutrition. So we can target this in a number of different ways, and this is basically what we're looking at. So this has been done with topotecan to turn on the paternal allele, and that was a great advance in what we know, and as far as a proof of discovery that this can be a viable target. The work that Jen Daly, as a matter of fact, Jen Daly is here in the audience, right over there, raise your hand. So she did the initial AAV work and published that paper that got Agilis so interested in some of the work that we're doing. I think the most important part about that work was we had done this in an adult mouse. So this was a mouse that had Angelman syndrome. We had injected the virus. We waited 5 weeks, and she saw this fantastic recovery. You can also look outside of the nucleus in places like the synapse, and there's been a lot of work done in that area as well. We've published a paper just recently on Reelin application that works on the synaptic connections there. Also work out of Eric Klan's lab, looking at the potassium ATPase, and then some other work with ampakins outside of the gene. So when all these therapeutics that we're talking about and companies, they're all targeting different areas of what we consider to be altered in Angelman syndrome. So one of the things, and Jody kind of touched about this, was the work that we're doing really kind of trying to develop new clinical outcome measures. And we think this is really important. And as Jody talked about earlier, coming up with clear outcome measures that we can use in future clinical trials is going to be very important, maybe even to the success of what we're trying to show. So how you define what you want to do and where you want to target and primary and secondary outcome measures is a big question that we're all trying to deal with, especially when designing new clinical trials. But the overarching question is which ones are going to be best for whatever therapeutic you're looking at. And so we're just trying to develop new outcome measures that are going to be less subjective, more objective, more quantitative, that you don't need somebody in there evaluating and saying, okay, we think we see a difference, or an evaluation of the parents saying, I think we see a difference. Something that we can go in, have the trial tested and say, yes, there's a definite difference there or not. And we really found this from the work that we did in the minocycline clinical trial using the Baileys and the Vineland, which in hindsight are really not very good tools to do an evaluation of Angelman syndrome. And what we found is that if you look at this this distribution, and this is what the average is on whatever you're looking at, and then 2 standard deviations away or 3 standard deviations away. And we use the term normal only because it's a normalization of the data, and that's what they would say this is where a typical child would be on any of those different types of measurements. But what we see with Angelman syndrome is there's a floor effect. So there's more than 2 standard deviations away from that mean. And what we see when we look at them in comparison with some of the measurements that you get from a typical child is that they fall so far outside that even an increase or even a significant increase, you don't really see it. So you may see observations with the child, but when you're trying to measure it and come up with a quantitative measure for this, it disappears. And so we live in science with things like statistics, where you say this is a significant difference using this particular test, and these tests is very difficult to show significance. So what we need to do is we need to develop new outcome measures, and that's what we've started to do. So one of the things that we're looking at is, can these measures be specific to Angelman syndrome? So one of the things we started looking at was the deficit that Angelman children have in verbal communication. Okay, so we saw this when we did the minocycline clinical trial, and we were looking at receptive and expressive types of communication. And we saw, again, this kind of floor effect, especially when you standardize the raw data and you look at that in comparison to typical children. So we started looking at using the Lena System. And the Lena System is actually this really interesting system that uses this microphone here. You just kind of press the button, it starts recording. You have all sorts of different clothing that's got a little pouch that you put it in, so it's non-obtrusive. and then you record over time. You can do this up to 24 hours, and then you plug it into the computer and it downloads everything that it's recorded. It's a sophisticated system that we're finding is trying to get the data out. But what you can do with this is you can isolate all the vocalization that's coming out of the patient, and you can quantify that on the computer. So we've actually done this in a number of patients. And this is one of the things that we came out with. So when you look at and graph these things out as far as things like control of phonation and canonical syllables, these are single syllables that are either vowels or consonants, and you look over a specific period of time, you can see that you get this distribution. And what you should actually see, because this is going from, this thing is working. I don't know how I'm supposed to point this. There we go. Wow. There's a delay on this, I apologize. What you're seeing is more primitive sounds to more mature sounds, and when you start talking about advanced forms, this is stringing words together. And this isn't just to show that Angelman children are different or your kids are different in how they communicate, but what it does do is it makes a profile that we can compare if we're giving a therapeutic. Does any of those things change? Does any of those different kind of parameters change? And this is a way that we can do it. We can quantify it. And it is completely non-subjective. Okay, so we can get the recordings off. The computer is going to tell us, does this change? Does this profile change to more of what we see with the typical child? See, the old-time slides, actually, I think I like better. There we go. And then it goes 3 ahead. So one of the things that we found, and one of the things that the speech pathologist found when she was doing a lot of these studies, and this is Ruth Barr at the University of South Florida, was that Angelman children, how they express themselves if they can't do it vocally is really through gesture. And so the quantification of gesture and looking at these gestures was a really interesting aspect of this. And because of the motor coordination defects that we see in Angelman syndrome, this becomes problematic as well. So they went through literally hundreds of hours of filming kids with Angelman syndrome to really look to see how that gesturing differs and how it differs from child to child and if they can actually quantitate that. And so we're actually pulling a lot of this data together to kind of show that there's not only changes in vocalization, but also in other nonverbal ways of communication. And we're bringing this up to publication hopefully in the next month. Wow. Okay, let me just go a little bit forward here. The second thing that we're looking at, and the closest thing that we have to getting the data together for publication, is work that we're doing with the gait analysis. So this here is what's called a gait mat. You can see where he's standing. It's looking at where his footfalls are. It's got pressure sensors in here. And what you can do is you can naturally have the patient walk back and forth on this, and it takes all these different measurements, not only on just the how they're putting their feet down, but also their cadence, how far apart their feet are, if they're pronate. So it takes all these things into consideration, and it's all computerized. So it quantifies all of that data. I can get to the next one. So the nice thing is that this has been used for a lot of different disorders. So for a lot of ataxic disorders, for example. So there's a lot of information about using this gait analysis to kind of quantitate all these different measurements in a person's gait. So we've already done some studies where it shows that one of the first things that we looked at was just width of stance. And we see that there's a significant difference for both left and right foot between Angelman patients and typical age-matched patients as well. But we also saw something interesting when we started looking at everything that had to do with the cadence and spacing and position of the feet when we looked at Angelman patients versus other patients. And this is all actually previously published data. One of the things that we found is that when we look at Angelman patients and we look at all those different parameters, you can actually compare these with children that have other intellectual disorders that are associated with gait disorder as well. And what we found is that when you take all those numbers into consideration, they don't look like those children. What they do look like is people that have ataxia. Okay, so it really kind of categorizes them a little bit differently. But again, we can get really specific numbers from this. This is all computer evaluated. We can supply a therapeutic, put them back on the gait mat, and now you're not trying to evaluate, well, is my child walking better? Are they moving around better? This will actually quantitate it and tell us whether we're seeing a significant difference or not. We have developed somewhat of a gait score on this. If we take all of the data that we get from a typical patient and we put it all together, it should equal basically 0. So we can take all the different parameters from the gait analysis that we do and come up with an overall gait index. And so not only can you see where that child falls within that standard bell curve of changes in gait, but we can also evaluate and say, does a therapeutic change that overall gait index? So we can look at it in a lot of different specific aspects and then in kind of a general gait index aspect as well. The other work that we're doing, and we're actually, this should have been put in for publication before I showed up here, was looking at overall synaptic function using the mouse model. So this has been a recurring theme, I think, with the new models that are coming out. It's going to be important to kind of establish this overall effect of synaptic function, and then we can move forward to do more work on trying to model what's happening in the synapse and synaptic connectivity later on in these other models. Let me look over here. One of the questions that we had was what is happening with synaptic function? So I've come up here and talked to you ad nauseum about things like synaptic plasticity and how connectivity between the neurons and how that increase in connectivity underlines learning and memory. So we've done this in our in vitro model where we can take a slice out of the brain, we can stimulate presynaptic, record postsynaptic, and we can see what that communication looks like. If we give high frequency stimulation, so if we can mimic somewhat like memory formation that's happening on a cellular level, we can see in the AS mouse model there's a deficit there. One of the questions that we had was alterations in proteins like ARC that are involved in receptor trafficking and how the synapses are communicating with each other. What was that underlying the deficit that we see in long-term potentiation? And looking at some of the other receptors and some of the other players in synaptic communication, the conclusion that we came out with was that perhaps what we were seeing was a depressed synapse. And I don't mean depression like they're depressed, but if you give low frequency stimulation, what you can do is you can actually reduce that connectivity, reduce that communication between two synaptic connections. This is important in learning and memory as well, and that function between potentiation and depression, that kind of yin and yang, is very important in aspects of learning and memory. So we were working on a hypothesis that actually what happens when you look at a non-AS mouse and you give high frequency stimulation, you can get enough potentiation to see this long-term effect. If you give saturating amounts, you get a ceiling or as much long-term potentiation as you can get. If you give low frequency stimulation or DHPG, which activates metabotropic glutamate receptors, you can get depression. If you do this multiple times, you can get the maximum amount of depression. What we were thinking was that perhaps what was happening in the Angelman syndrome mouse, that instead of just giving high frequency stimulation and not getting as much long-term potentiation, that what we were actually seeing was a change in the baseline of where you were starting from. So if the synapse is already at a depressed state, You give high-frequency stimulation, you don't get as much long-term potentiation. And we've already shown that you can saturate this and get to the same amount of LTP as you can with a typical mouse. What we found, though, is that if we give multiple trains of low-frequency stimulation and also DHPG, so make it as depressed as possible, what happens is you can get to the same amount of depression as a typical wild-type mouse. Go just a little bit further. And so what we're actually seeing is that the defect in long-term potentiation, or the potentiated state, is the exact same thing as we see the problem that we see with the depressive state. So this does give credence to this model of overall reduction of synaptic connectivity or communication. So we've talked about this before, where we say if you have an alteration in synaptic communication between two neurons, it's not a big deal. If you do that globally throughout the brain, then you start having a syndrome. And this can affect a lot of different areas of the brain, so it's going to come out as a lot of different symptoms: changes in sleep, changes in hunger, changes in seizure ability. And this is kind of where we come up with the idea of a global therapeutic as well. So can we treat all the synaptic connections? So if there is no change in the depressive state or the resting state of this particular neuron, the problem is in communication either on the depressive state or on the excitatory state, that a global therapeutic would make sense. Okay, so you can affect things globally in the brain, and it appears that communication is really where the problem lies. The last thing that I want to talk about is something that we've been working on with Agilis, talking about, or at least looking at UBE3A activity and enzymatic activity. So you've seen these kind of slides before, where you're looking at UBE3A. What UBE3A does, it targets proteins for ubiquination, and then they go through the proteasomic pathway and get degraded. Then the ubiquitins get released and then brought back into this cycle. And this cycle happens at a certain rate, right? So like any enzyme that you look at, the amount of ubiquitins that this UBE3A can put onto this target is regulated. It will change with things like temperature, it'll change with the amount of substrate that you have, or even the amount of target that you have. So this idea of turning on a paternal gene or doing gene therapy, or anything that would have to do with alterations in UBE3A, one of the questions is, well, even if it's there, is it still working at the same effect? Okay? If you're trying to get a Ferrari, yeah, you got a car there, but is it going to be like a Ferrari? And so what we've started to do is develop a test for this and do enzymatic activity tests, where we have two different ways to assay this. We can see a clear difference between the tissue that we get from Angelman Syndrome and then from wild-type tissue. We can look at that enzymatic activity, and what we can also do is we can compare this with anything that we do with gene therapy, anything that we do with enzyme that's coming off of the paternal gene. We can say, well, how well is this working? Is it being less active? That means that the curve would shift to the right here, or is it being more active? It could actually shift to the left. Alterations in that activity is going to have a lot to do. Somebody talked about, how much is too much UBE3A? Well, it's not only just expression, but it's how active that enzyme is. So even if you get up to normal levels of UBE3A, the question is it still doing what it's supposed to be doing even at that equal level? Dr. Burdine talked earlier about this regulation of UBE3A and how alterations in what they found is phosphorylation in a specific site can actually change that regulation. It can hang around a little bit longer, but it's kind of less active. So what does that mean for future therapeutics? Well, at least now we have a tool that we can measure it, we can quantify it, and we can see whether specific therapeutics, such like gene therapy or protein replacement therapy, is actually replacing the protein with something that works as well or better than what's there endogenously. So just very quickly, I know I'm probably going a little bit over, just very quickly I want to acknowledge some of the people that were involved in this. Dr. Barr is the speech pathologist that did all the work with the Lena System. Like I say, we've just submitted that for publication, so hopefully that will get out, and you can kind of look at all the results and the conclusions that we've come out with that. Irina Filanova did a lot of the work on the synaptic function in depression. Joe Greco and Stephanie Charlone, which you probably have seen before and met before. Stephanie did all the work that was shown earlier with the ketone ester, and Joe's done a lot of work trying to figure out these new outcome measures. Dr. Lee and Christine Lemens have worked on the UBE3A assay. And then Justin Trotter, I just have in here. I don't even know why his picture's on there. He's an old graduate student that's now working at Princeton. But with that, I will take any quick questions.